Good day, students. You are welcome to today's episode of our lecture video. Today, we are going to be discussing about tips for successful biochemistry practical exams. So, over the last few weeks, these are the practicals we covered. We discussed about spectrophotometry, introduction to colorimetry, and we discussed about introduction to absorption spectrum, preparation and uses of standard curve, qualitative test for carbohydrates, preliminary test for lipids, determination of iodine number, qualitative test for amino acids, and lastly, we discuss about quantitative estimation of amino acid and proteins by formula titration. So I want you to remember these important points. And for each of the practicals, there are things that you are expected to remember so that you have a maximum point in your exams and tests. So under introduction to colorimetry, you are expected to remember the concept of spectrophotometry and the basic principle behind absorption of light by certain compounds. You are expected to take note of Beer's law and the formula that says absorbance is directly proportional to concentration, and which says you can now simplify the expression as absorbance is equals to molar extinction coefficient multiplied by concentration and multiplied by the path length. You're expected to take note of Lambert's law and also the combination of the two laws, Beer-Lambert's law. You are expected to also understand the differences between colorimeter and spectrophotometer. So these are things that you are expected to understand at the end of this practical under introduction to colorimetry. So under introduction to the absorption spectrum, you are expected to understand the meaning of absorption spectrum and also the absorption spectrum of some certain macromolecules like proteins, nucleic acid, and some compounds, uric acid, NAD plus NADH, NADP plus NADPH, hemoglobin and its modified forms. And also you are to take note of the differences between absorption spectrum and standard curve, and also the plots of absorbance against concentration. You are expected to understand that. And under preparation and uses of standard curve, you are expected to understand what a standard curve is. Standard curve and calibration curve, they mean the same thing. And what is the plot that is absorbance against concentration? Okay, so for standard curve is a plot of absorbance against concentration. You are expected to understand that. And absorption spectrum is a plot of absorbance against wavelength. And also you are expected to know the components of the biorate reagent and also how to carry out the serial dilution. So under qualitative test for carbohydrate, you are expected to understand the differences between qualitative and quantitative test and the classification of carbohydrate and the qualitative test that we carried out, Molish test, Antron test, iodine test, Barford test, Selwanov test, Bell's test, and also the reagent involved, their function, and the principle of each test, and also the color you observe. So under this qualitative test, you are expected to take note for each of the tests. What are the reagents that are involved in the test? And what is the function for each of the reagent that is involved? And also, what is the principle behind that particular test? Is it a general test for carbohydrate or a specific test for a certain group of carbohydrate? And what is the color that you are going to observe? These are things that you are to take note of. So under preliminary test for lipids, you are expected to understand the meaning of lipids and the classes of lipids, under which we discuss about triacyl glycerols, which are the neutral fats. You should understand saturated and unsaturated fats and their examples. You should understand the test that you carry out, test for the presence of double bond and test for free fatty acid. You should understand the concept of rancidity, the types of rancidity, the causes, and also you should be able to understand what acid value is. So under determination of iodine number, you should be able to define iodine number of lipid and what is the significance of the iodine number, and why is bromine used instead of iodine in DAMS method, and what is DAMS reagent and how do you handle DAMS reagent. In case of spillage, how do you take care of the spillage, and what are the components of DAM reagent, and also how do you determine the iodine equivalent, and how do you calculate the iodine equivalent. So under qualitative test for amino acid, you should understand the 20 common amino acids that are found in proteins, and what are they, what is the general structure of amino acid, and what is the principle behind each of the tests that you carry out under amino acid, qualitative test for amino acid, and what is the color change, and what are the reagents involved. Okay. So, and under quantitative estimation of amino acid and proteins by formal titration, you should understand what a monoamino monocarboxylic amino acid is, and also what happens if you have that amino acid solution, an acid is added to the amino acid solution, or a base is added to the amino acid solution, what happens, and what are the reagents involved. Okay, and what is the endpoint, and also what are the calculations that are involved. So let's quickly look at some few questions and answers. So question 1 says define iodine number. So what's iodine number? Iodine number is of lipid sample is defined as the number of grams of iodine absorbed by 100 grams of lipid. And we can say what is acid value, and acid value is the number of milligrams of potassium hydroxide required to neutralize free fatty acid present in 1 gram of fat. Then what are the components of DAMS reagent? We should know that DAMS reagent consists of bromine, pyridine, sulfuric acid, and glacial acetic acid. And when the fourth question, it says, why is formaldehyde neutralized with sodium hydroxide during formal titration? So it's just simply to remove traces of free formic acid. That is why sodium hydroxide is used to neutralize the formaldehyde. And why is formaldehyde added to the amino acid solution during formal titration? And this one is to the effect of adding formaldehyde is to shift the pKa of the amino group to a lower value, making it stronger acid, and the group can now be titrated with sodium hydroxide using phenolphthalein indicator. So these are some of the few questions that you are going to be expecting. These are the patterns of the questions that generally you should be expecting. And if you look at it, you'll see that for each of the tests that we carry out, or each of the practical that we carry out, you should be able to remember each component of the reagent that you use, and what is the function of each of the components of that reagent. Okay, and also if a principle is involved, what is the principle for that particular test? You should be able to know that, so that at the end of the day, if a question is asked, these are just some few questions that we sample out. So if generally something like this can just come out, so you can see that at random, you can be able to answer those questions. So for each of the tests, if there is a principle involved, make sure that you know the principle of that test, and make sure that you understand the reagent that I use and what is the function of each of the reagent. And also, if there is color that is involved, what is the color after the reaction? Okay, what is the color change? What is the color at the end of the reaction? Okay, so and if there is calculation, how do you calculate? How do you do that calculation? Please sit down and understand all these things. Biochemistry practical generally is not difficult. It's easier to pass. It's very easy to pass, and you can pass it with good grade if you pay attention. So if you remember, there are things that you are supposed to do. But the first thing is to make sure that you attend the lab, you attend the pre-lab. So that's the first thing we do in the morning, pre-lab, before we start lab. That pre-lab is very important. A lot of information will be shared with you during the pre-lab on how to carry out the experiment. And after the pre-lab, make sure you participate in the experiment. So after participating in the experiment, if a question is asked with regards to the experiment, you can be able to remember because you participated in the experiment. And also read your manual. The manual has a lot of information that you require to pass. All these questions, if you can see, they are from, all the answers of these questions, they are directly from the manual. So you read the manual, also read and understand the manual, okay? And also try to remember all the important points that we pointed out in the during the pre-lab and also during our tutorials. So it means all the videos that we highlighted, all the videos that we made during the past few weeks, you are required to go back, sit down, and digest the content. Okay, then you read your manual and also check your report. Where you have issues, you can always ask questions. I think when you add up all this information, you are good to go. You are going to answer your test questions effectively and also your exams questions. I think you can stop here. If you have any questions, you can always drop it at the comment section, or you can directly contact. Thank you for watching. See you next.